Hi student, this is in this whatever I am going to give you the lesson. In this lesson, he has given last year 2025 two neat bits came from this only. That is the pistil, Megasporogenesis or ovule and embryo sac. From this one he asked two questions. I will show those things also. And up to now in the 2020 he asked two and 23 one question. Total he has asked the three questions up to now, and it is most important topic in the sexual reproduction of the flowering plants. So you have, why he is giving this question means most the bits which can be convert means they will be the bits can be created, bits can be created in this such type of topic it is, but it is simple one. The thing is the pistil, megasporogenesis or ovule and embryo sac all will come under the female sex or part of the flower, female sex part of the flower. And next, whenever we are, and it is called as female sex part is it is called as pistil. This is the new word. Last year morphology he called it carpel. Both are same. Difference is there. What is the difference? Finally, I will tell. By in this time, during this time, think over it. Any difference is there or not? Now you see here, the flowers used to have different means. It may be having one pistil or many. If it is having the one pistil, it is called as a monocarpellary. And if it is having many, multicarpellary. Monocarpellary means one pistil, multicarpellary means many pistils used to present on the thalamus. And if the carpels which are present on the thalamus, more than one, it is multicarpellary, they may be fused like this. They will be fused like this and for appears like one pistil, appears like only one. That is called as a syncarpous. And if they are present, all these are the carpels like this different they are not fusing, then it is called apocarpus. This is very important for need. Apocarpus, syncarpous. And then when we are seeing, now we will see the what are the parts present in the pistil. So in the pistil consists of three parts, that is stigma, style, ovary. Stigma, style and ovary. You see here. Stigma, style, ovary. And these are the parts of the pistil. Pistil, when we are, this is also called as carpel also. That why it is the difference in the last I will tell. So the stigma is the terminal part. And it is like platform. What it is, it is give the it works as a platform for the landing of the pollen grain on that, because it has to receive pollen grain which is the male gametophyte, and it is to produce two gametes. It will come in the fertilization, and it will be where the pollen grains will be land on the stigma. It is works as a platform, and there is a slender and slender filament like structure used to present in between the stigma and ovary, that is called as style. Style is a elongated part which is to present between the stigma and ovary. And what it is to do in the anther also I told, anther or this style, they used to facilitate receiving of the pollen grains here, there for the pollination they used to do. They will be helpful for the pollination. That is the different. I will discuss those things lastly. Now you see here, and the third part which is basal part attached to the thalamus, it is bulged one, means swollen one like this, and this is called as a ovary. ovary. So this is the main part where the ovules used to present, ovary. And which part is attached to the thalamus? Ovary. This ovary is to have a chamber inside, that is cavity. That is called as ovarian cavity. Whenever the chamber is there, that will be filled with the tissue called as placenta. The placenta cells, some of the placenta cells used to give rise the ovule. That is the important, because eucellus cells used to give rise the female gametophyte. Placenta cells used to give rise the megasporangium, because ovule is called as megasporangium. So ovules are born from the tissue, some of the cells of placenta. And next, whenever we are seeing the ovule, all these three parts used to contain, and these are the works. Now, whenever we are seeing here, the same thing. Pistil consists three parts: stigma, pollen, grain, landing platform. Style is the elongated connection between the stigma and ovary. All these are written by me only, but the thing is, I converted into the printing matter way on this board, because easily you can see. Now, the ovary is the bulged basal part of pistil. It contains ovarian cavity, which fill. This is the which which will be filled. Because of this some problem, it came like this, with the placenta tissue. The placenta tissue, where it is present? In the ovarian cavity. And now, and it contains of ovarian cavity, which filled with the placenta tissue. Now we will see the Megasporogenesis. Now these things, one more I want to show you. Here, this diagram he has given in the textbook, NCERT textbook. Now here you see, I will tell you, now the time came. That is, on the thalamus, ovary is there, and style, stigma. Okay, but it is what he has written there, that is the hibiscus, means China rose he has given. This is how many carpels are present in this one? How many? 5 are there, because the stigma is rated, that Malvaceae you will read. And 5 carpels are present in this one, but he is calling it as entire pistil. This is a pistil. Means united things, either it is maybe one or many, whatever is there, then it is called as pistil. If it is having the style, stigma and ovary, then it is called as carpel. How many? This how we will describe? Pentacarpellary, syncarpous, they are fused together, and pentalocular. We used to tell, means pentacarpellary, syncarpous, but it is called as pistil only. Remember it. Because why I am telling this much detailedly, he will ask the questions by the, he can create in the need. For that one I am telling. And this is also pistil. What is this? Papaver. How many? All these are the carpels. They are fused together, stigmas. And it is also, he told in the under that one here, what he wrote, you see, it is also pistil. It is apocarpus, it is syncarpous, it is apocarpus. All the carpels are free. For that one he has written carpel, he has written there. And under that one he has not written about the pistil. Carpels are free. Apocarpus condition, pistil is not applied. Syncarpous condition, it is applied. And single carpel also it is applied as the carpel or pistil. This is the difference. Try to read it. And next, whenever we are seeing, these are the things he has given. This is the papaver, it is mycelia. Examples are very important, remember it. Because you have to see now and then, he will give the examples for matching or some other thing. Now you see here, the Megasporogenesis. So what we told, Megasporogenesis, what is called? Ovule is called as Megasporogenesis. This is the ovule structure given in the NCERT book. Now I am going to explain you all the things because it is the NCERT class only. And now whenever you are seeing the Megasporogenesis, Megasporogenesis is, we used to call it, ovule is called as Megasporogenesis. But in the male gametophyte, this is the anther. 4 sporangia used to present, many spores will be produced. Male gametophyte, that is the pollen grape. But here it is one ovule, which is called as a Megasporogenesis, and it used to give rise only one male gametophyte. But whereas in the anther, the microsporogenesis, many pollen grains used to come, but here it is to produce only one. Or the female gametophyte. Now you see one by one. Here it is to have, from where it is they are born, ovules are born from, already I told, that is from the placenta cells. Placenta cells used to give rise, some of the cells used to take the divisions and forms the ovule. So from the placenta, one stalk-like structure used to come and it will attach to the ovule. That is called as a funicle, stalk of ovule. And whenever the stalk is directly coming and attaching to the ovule, there will be a joint. That joint is called as hilum. That is the funicle ovule junction. It is wrongly also he has given some seen. That is always it won't be the hilum, it won't be present at the bottom of the ovule because it is an anatropous ovule. Arthropous ovule is vertical. It in the gymnosperms it is to present. Arthropous, the funicle, whatever is there, it will come under this. So vertically on single line. But here in the anatropous, the funicle and ovule, they are parallel. They are in the parallel condition. Anatropous also is written there. Remember, it is anatropous ovule. And next, whenever we are seeing here, the ovule is covered by the protective layers called integuments. So, called as integuments. Integuments used to give protection to the ovule. They may be one or many. And inside the integuments used to leave some pore from the one side at the terminal side or somewhere it is to leave. That is called as a micropyle. Then wherever it is leaving the pore, it is micropyle. Where are the exine used to leave the pore in the pollen grain. Like that he can give. The pore which is to present in the pollen grain is, and pore which is to present in the ovule is micropyle. And micropyle pore will be useful for the entry of the pollen tube. That is the thing. And the next will be the inside the integument, there will be a tissue naturally, because in the inside the ovary placenta tissue is there, inside the ovule tissue will be there. That tissue is called as nucellus. So nucellus tissue, very important tissue, and the tissue is ovule side, basal side, the tissue inside the tissue, the ovule, they are ovule rich of food. This is the inside the ovule, the tissue inside the ovule, because something problem is there. So inside the ovule, whatever the tissue is there, that is called as nucellus tissue. It is stored food material used to present that one, because growing embryo or embryo sac, and for later on for embryo, it will be used. Food material reserve will be in Megasporogenesis, where the food material used to reserve will be there, means nucellus it is. Remember, like that questions will come. And next, whenever we are seeing the nucellus, wherever the pore is left in the integuments, that is called as micropyle. That end is called a micropylar. That end is called as a micropylar. Opposite to the micropylar end, that is the base or somewhere, we don't, because it is parallel. In the orthotopos it is vertical, but opposite to the micropylar end, it is called as the chalaja. Wherever the micropyle end is there, opposite to that, it is called as chalaja. Chalaja pole, micropyle or pole. And next, inside the nucellus, female gametophyte will develop. Female gametophyte is developing where? Inside the nucellus cells. One of the cell will produce the female gametophyte. Now, Megasporogenesis. What we told, Megasporogenesis. Megasporogenesis is over. What is the Megasporogenesis? It is to have the panicle and the hilum and the integuments, and it will have the micropyle inside the nucellus cells. Inside the nucellus cells, it will have the female gametophyte. Now you see Megasporogenesis. How the megaspore will be formed? Megaspore, their microspore formation. Here it is called as a megaspore formation. So formation of megaspore from megaspore mother cell. This is the cell. It came wrongly whenever I am converting, because I am not familiar with this board. Now I will become familiar. So don't feel bad. So fallen mother cell, MMC, megaspore mother cell it is, and called as a megaspore mother cell, used to form the formation of megaspore from megaspore mother cell, called megasporogenesis. Megaspore mother cell used to give rise it is to megaspores. So here also meiotic division used to take place, 4 will be formed. That only he has given. Now you see, one of the nucellar cell, nucellar cell, whatever the nucellar cell, at the micropylar end, differentiate as megaspore mother cell, MMC, megaspore mother cell. This is the micropylar end. Micropylar end, wherever it is there, one of the cell will become very big, differentiate, big size, and it will have the cover also, that is not mentioned, nucellus cover. Megaspore mother cell, it will be formed. This megaspore mother cell, what it will do? Megaspore mother cell, this is a diploid one. It has to take the meiotic division to form the haploid megaspore source, megaspores, 4 megaspores. But here he has given some critically, he has given, that is, the megaspore mother cell has formed the 2 cells, which is called as a megaspore dyad. What is this? Dyad, where it came? Dyad came in the meiosis. After meiosis 1, 2 cells used to form, which are called as dyad. That word is, this fellow has used this. So it is also having some importance, I will tell you. That is, the dyad cells, megaspore dyad cells used to form after meiosis 1. And next, this will take meiosis, that is a Megaspore dyad will take one more mitotic division. Means meiosis 2 is nothing like mitotic division only, and forms 4 spores. So reduction division forms 4 spores of megaspores, and they are the megaspore tetrad. Yes, given. What microspore also used to form tetrad. Microspore tetrad. Here megaspore tetrad. What is the difference? Microspore tetrad is, it will be the cluster, but it is linear. It is that only I shown linear formation. This you can twist and give. For that only I am going in that much deeply. So this the dyad cell will form after meiosis 1, and in this way 4 megaspore tetrad is formed. Megaspores are formed from where? From the megaspore mother cell. Megaspore mother cell, how it formed? One of the nucellus cell, which is at the micropylar end, used to differentiate and forms the megaspore mother cell, and it will take the meiosis, and after meiosis 1 it used to form the megaspore dyad, means 2 cells, and after that it will become 4 cells. They are the megaspore tetrad, which are linear. Now we'll see female gametophyte. Here they falling grain, because what I am telling, it is need orientation, and if you are, whenever I am comparing, you may get confused, but don't get confused. You see once more, because it is most useful for the NEET, and appearing, whoever are appearing for the 27 board exam also, it is important. In most of the flowering plants, one of the megaspore forms a functional, one of the megaspore only will be functional. How many are formed? 4. Out of that one, only one will become functional. And now whenever we are seeing, out of 4 megaspores, one will be functional, and the functional megaspore used to take further divisions and forms the female gametophyte. That only he is going to explain here. Now in the textbook he has given all the things. It is called as mono, and out of 4, 3 are degenerating, one only becoming functional and forming the female gametophyte. So this type of development is called as monosporic. Only one spore will be developed as a female gametophyte, whereas in the microspore tetrad all will form male gametophytes, but here 3 will be degenerated megaspore, so only one will be functional, that will form the female gametophyte. Now you see here how the female gametophyte is forming. He has written in the textbook, he has given. The thing is here, you see the whatever the functional megaspore will divide, it will take the mitotic divisions and forms the free nuclear. First mitotic division will be taken in the functional megaspore and forms the 2 nuclei, and it is not following the cytokinesis, karyokinesis only will be. And first, second, third, 3 times it is to divide mitotically, only karyokinesis. In the first, after first this one, whenever it has taken first division, mitotic division, it used to form 2 nuclei, and both the nuclei used to move to the opposite poles. They used to move to the opposite poles, and after reaching the opposite poles, they will do the one more mitotic division, and each one used to form the 2 means 4 nucleate embryo sac. It is called, and first one he has written that is a 2 nucleate embryo sac. Okay. Now each pole, how many are there? 2 are there. Again one more mitotic division will be taken and it will become the, at each pole 4 free nuclei used to present. Here, up to formation of the 8 nuclei, it is taking the mitotic division. The free nuclear divisions without following the cytokinesis, it is having a technical word given in the mitosis of the cell division. What is that word? Syncytium. Remember, note down. Syncytium. That there he has given coconut water. Liquid endosperm is free nuclei used to present this called. Not only that one, that a formation of the embryo sac also, it used to follow syncytium. And next, free nuclear divisions, it is syncytium. Now the thing is, 2 nucleate condition has formed from the functional cell. Both they migrated to opposite poles, and it is it is called as a 2 nucleate embryo sac, and it will become each pole, the nucleus will take again mitotic division, forms 2 cells, 2 nuclei, and again one more mitotic is used to form 4 nuclei at the opposite pole. And this is the thing is free nuclear divisions. Then it will start the cellular formation. Up to now it is a free nuclear. Now it will become cellular formation. How? Only 6 cells will be formed. Cytokinesis will be taken when, he will ask. After forming of the 8 free nuclear condition, cytokinesis will take, and they used to form the cell walls and form the cells. So how the cells are? 6 cells are present in the opposite poles. 6 cells are formed in the opposite poles. And chalagel and whatever it is there, the 3 cells are called as antipodals. The chalagel side 3 cells are called as antipodals, and micropylar and 3 cells are called as, they are called as egg apparatus. They are called as egg apparatus. And now whenever we are seeing, now 2 nuclei are left because 4 had formed, 3 are participated in the formation of the cells. The 2 cells, 2 nuclei which are present, they will be present in this one. This entire thing is called as central cell. Now you tell me, which cell is the biggest cell in the female gametophyte? Central cell, because they are present. This entire thing is central cell. This is also neat with. So this entire cell is a central cell. And now chalagel and 3 cells are called antipodals, and micropylar and 3 cells are called as egg apparatus. Egg apparatus consists of the 3 cells already we told. 1 cell is the egg and 2 cells are the synergids. They are called as synergids. So in the matured embryo sac and female gametophyte we are seeing, it is to have this micropylar, means reversely has given micropylar, and what are present? Egg apparatus are present. They are the 2 synergids, 1 egg. And these are the centra, the polar nuclei, means which left from here, and this one, this and this is called as polar nuclei. And here he has mentioned one more point, that polar nuclei are near to, just above to the egg apparatus. Most important point, because he has written in the text also. Why? Because it must receive a male gamete. For that one, it will be very near to the egg apparatus, far to the antipodals. Clearly he has written in the textbook. Whatever he mentioned in the textbook, they are very, very important for me. Generally, students used to neglect those things. They will feel very bad. They will lose the marks. And one more thing, the synergids, whatever are there, is the micropylar end at the lower, attached to the synergids which are attached to the base of the embryo sac. They will form some thickenings, like finger-like thickenings like this. And these thickenings are called as filiform operators. Filiform operators are having some function, because whenever the pollen tube is entering into the embryo sac, it will guide, means whenever it is like this, the pollen tube go like this, it will give the way, it will guide. The pollen tube will be guided into where? That also you will ask. Into the synergid, clearly mentioned. Not into the egg, not into the central cell. The filiform operators used to guide the pollen tube which entered into the embryo sac into which cell? Clearly given in the text. Read it properly. And it used to guide the pollen tube into synergid. What happened to the synergid, that we will discuss in the double fertilization, triple fusion. So here now what happened? This is one of the megaspore which is formed by the taking of the meiosis of the megaspore mother cell. It has formed the two cells, dyad cells, and two cells again taken mitosis division, and it forms the, means meiosis 2 division forms the 4 megaspores. Out of 4 megaspore, one will be functional, and functional megaspore used to form the polymer, that is the female gametophyte, by taking the free nuclear divisions 1, 2, 3 that we discussed, and later on it used to form 3 cells from the chalazal end and 3 cells to the micropylar end, and micropylar end cells are called as agapparatus, and chalazal side 3 cells are called as antipodals. And next, whenever this condition is called as 8 nucleate, 1, 2, 3, 4, 5, 6, 7, 8. 8 nucleate. Cells are 3 plus 3, 6, this under the one cell. And this is the polar nuclei. We used to present, 2 nuclei used to present in the single cell. That is a central cell. Central cell contains of 2 nuclei, haploid nuclei, both are. And one more thing, if people, when they will fuse, some people used to say it is diploid. No, it is a haploid cell only, because 2 nuclei are present, 2 are haploid. When they will fuse before the fertilization, first fertilization means, if you know that neat going people, they know. Double fertilization, triple fusion. First fertilization will be 2 gametes will come from the male gametophyte, and that it is to one will be first fused with the egg. First fertilization. Second fertilization will be taken place with the polar nuclei. So here, when the fertilization is over, then both the nuclei used to fuse just before the fertilization. Up to that, in the matured condition of the embryo sac, will have the 2 haploid polar nuclei. Remember it, because he can give the, already last year 2 questions he has given. Out of that one, this he asked that thing from the starting. He asked from the megaspore mother cell to the formation of the female gametophyte, how many mitotic divisions are taken, how many meiotic divisions has taken. It has taken only 1 meiotic division in the megaspore mother cell, and mitosis cell division will be taken, mitotic cell division will be taken here 1, 2, 3 divisions. That is the answer. And the next question is assertion and reason. In the assertion and reason, what he has given, assertion is, that is assertion and reason, and what the reason is wrong, he has wrongly has given, that is a 8 nucleate and 7 celled condition. It is, assertion is a it is 8 nucleate and 7 cell. That because it is already shown. So embryo sac contains of the 8 nucleate and 7 cells it is, and synergids are having the polar nuclei. Polar nuclei are present in the central cell. That is wrongly he has given. And 2023 he has given one question: sequence. How the megaspore mother cell to how dyad and megaspore to the again all these formation sequence he has given. And 2020 he asked about the ilum, means junction. And 2022 when 2021 question he has given, that is out of the following which are present haploid two generations, one in other generation, means generations means haploid and diploid. For example, here whenever we are seeing this where it is present embryo sac, initially it is to present inside the nucellus. Nucellus is the diploid, it is the haploid. In the same way that that male gametophyte, what is pollen grain in the anther. Anther, whatever the cells, other cells are the diploid, pollen grains are haploid. That is the thing, same thing. He has given the pollen grain, but he cannot give this time the also. This is the haploid, and where it is present? In the nucellus. Nucellus is diploid. So the diploid generation, the haploid generation, both are present in the Megasporogenesis. These are the things. And next, micropylarine forms the techniques that I already discussed. And these are the things about the, actually it is not so difficult. But the thing is, you have to read it properly and understand. If you follow this, whatever the video I done, you will give complete any type of the question he gives, you can give the answer. And in the 2027 examination also, if it comes, CBS exam, you can give the answer. So if you like this video, please give the like mark, and similar type of the videos will come, all this about this chapter and next all the chapters. So to follow all those things, please subscribe the channel. Thank you.